Hello, guys. Can you hear me?
Yeah.
Yes, sir.
All right. I have to use a different computer this morning because my laptop, for some reason, keeps crashing. I don't know what the issue is. It's overheating. So, it's Monday, 11 o'clock. So we have a test coming up on Wednesday. We're finishing chapter 5, protein transport. So we'll talk more about how the oxygen binds to hemoglobin and how that binding is regulated in such a way that as much as possible will bind in the lungs and as much as possible will be released in the tissue, right? So we're going to learn about other tricks that the living systems have, or us humans have adopted to regulate that process. Now, as far as the exam, I know you probably have lots of questions about that. So I gave you some pages to review, right? So again, ideally, these are the pages, ideally, these would be the pages that you have already read after lectures, right? As the semester went on. So you just need to review those. But I just sent you those, so because that should be the focus for your review, all right? So, which kind of tells you that most likely should be expecting questions related to those pages. So the exam is going to be going to have 30 questions. I'm going to give you 55 minutes. So it's not a whole class exam. We don't need such a long exam because most questions, as you will find, there's not so much math. In fact, there's very little math, a couple problems, maybe just one with little math. So most questions will be sort of yes or no type, or pick the all multiple choice, just pick the right answer, right? So 55 minutes should be plenty for you to answer those 30 questions. In fact, most of you will probably finish early. I don't want to give you the entire an hour and 20 minutes because that sort of opens the door for you guys to have extra time to start Googling things, which I don't want to happen, right? And those of you who take the exam honestly, I don't want you to worry about others Googling the answers in this class. So what else? So, and also when I put the exam together, it took me actually a long time. So obviously these questions cannot be easily Googled. So it's not just like quizzing your knowledge on things like what type of secondary structures are there, or it's mostly for you to think about. So to use your knowledge, right, and conceptually to think of an answer. Like the most common one that we've been talking about over and over is if you have a small peptide and or a mixture of amino acids, which amino acid will be eluted first from a cation exchange column. So stuff like that. So you should be expecting a problem like that. And obviously you cannot Google that because this is something for you to think about. Do you have any questions about the test?
Are we going to be on Zoom when we take the exam?
Well, Zoom will be, the exam is kind of personal, right? Whereas Zoom is kind of open to everybody. I mean, I'll be on my email. How about that? So I'll be.
Okay. The reason I ask is because I don't know if it's my computer, but when I have multiple pages open while I'm on Zoom, it is really slow and doesn't work.
Yeah, so it's good. So it's good.
Okay.
It will not be on Zoom, right? So just open Canvas, go to quizzes. You will find it says test 1 and start working on it. and you will have 55 minutes, and there'll be a timer there telling you how much time you have left. And if you have not submitted the exam yourself by the end of the 55 minutes, it will submit it for you. It's a very nice, friendly system. So yeah, you don't need to be on Zoom, but you can send me an email. I'll be on my email as you guys are taking the test. So.
Will the test only be during class time, or do we have like certain time during the day to do it?
Class time. So obviously, you know, I can't really dedicate the whole day or 2 days for you to spread it all over a period of time for you to take tests at different times. You know, there are over 65 students in class. So during class time, yes, so there are a couple students who need extended time through the Office of Disability. So they will take it at a different time.
Will we be able to skip around in the exam, or will it be like you have to answer the question before you can go on to the next?
Yes. So again, I arranged, again, many professors at Texas State and other universities use various proctoring services. So Texas State doesn't really offer that because many of them don't really work very well, and they just increase the technological problems for you guys. You know, you have to have 2 computers or 2 whatever camera and so on and so forth, and there are just increases the number of things that can go wrong. And so that's why instead of doing that, I just designed the questions in such a way that it's going to be hard for you two guys to cheat. It's going to be hard to and so to answer your question, you will have to answer one question at a time. Yes. And they're going to be randomized. So like it's going to be very hard for you to get in to do it in groups, for example, or to do 2 people at the same time, because each two people will have different sequence for these questions. So once you submit a question, you will not be able to come back to it until the exam is submitted. Once the exam is submitted, then you will have an opportunity to view all your answers and correct answers. Right. There is actually there are a couple math problems. Well, I can think of one, but just in case I'll tell you 2. But I think it's just one math problem. But it's very straightforward. It's kind of related to one of your sapling problems. So it should not present much. That's why I told you guys sapling is a great way to learn, and I also took advantage of sapling quite liberally when I prepared this exam. So many questions are similar to problems in sapling. So, but bring have a small calculator with you, or you can use one on your computer, obviously. It doesn't need to have log values or anything, just simple arithmetic calculator.
Dr. Kornienko, I have a question.
Uh-huh.
So you mentioned earlier that we would not be able to go back on, like, whenever we do the question, it'll kind of take us to the next one without having to go back. So if we have a question that we need to flag for review, we won't be able, just to clarify, we won't be able to go back and revisit that question.
Correct. So once you submit the question, you cannot go back. That's it.
Okay, thank you.
Mm-hmm. So yeah, everything is just to minimize, and minimize opportunities for cheaters, right? So it's, I think I've done my as much as I could in that respect. So anything
It
else?
sounds fair.
Yeah. Sorry.
I said, it sounds fair.
Yeah.
It sounds good.
Yeah, but as I mentioned to you also, there is really no need. I mean, other than if you talk to somebody and this person tells you, and you start discussing questions, there is really no need. If you work by yourself, there is really no need for you to go back. I mean, you spend some time. So what you have, in general, you have 2 minutes on average per question, but some questions you will see you'll probably spend a few seconds. So you'll have minutes on each question. So there's really no need for you to go back, right? So you thought about the question, you came up with the answer, and you clicked on it. You click what you think is the right answer. And the questions are not they also randomized, right? So they're not like by chapters, just like questions from chapter 1, then questions from chapter 2. They're all over the place. Which I learned that Canvas is not such a friendly platform to do that. I had to figure it out how to do it actually. Yes, 30 questions. Anything else? Well, if you have general questions, put it in discussion. If you have personal questions, you can email me. And again, guys, I will, if you have technological issues, I obviously can understand that. We're not fully immune to those, right? Our internet at our homes cannot be fully 100% reliable, but it is in our power to do everything possible to avoid these technological issues, right? If we have a unreliable internet at home, then maybe it makes more sense to take a test on campus, right? If you live close to campus, where the internet is quite stable. So all right. If there are no more questions, let's go to the material. We're going to finish chapter 5. So just to remind you, we looked at this before, but it would make sense to go back and because our discussion will start with this. Can everybody see my screen?
Yes.
Yeah. All right, thank you. So here there is a protein with 2 subunits, and it shows the cooperativity. That's how cooperativity works, right? So you have a binding site on this subunit, and you have a binding site on this subunit. And you can see at this moment, so let's say the protein is not quite prepared to receive a ligand, but once it does receive a ligand, let's say one of the sites accepts a ligand here, and then the lid comes down, there's some kind of, let's say there is very nice interaction here, right? So the lid comes down and closes, then this will change the conformation. You can see that this lid was actually wide open, right? And now it's partially closed. So now this binding site is much more likely to bind to the ligand than it was originally. And it's all has become possible because the first ligand has bound. Okay, and then the second ligand binds and this lid closes completely. So this is an example of positive cooperativity, right? When binding of one ligand facilitates the binding of the subsequent ligands. And so we talked about this in the context of hemoglobin, right? And so we'll talk about this in a moment again. So we briefly chatted about this. And so such proteins which exhibit this kind of cooperativity, basically it's what's known as a special case of allosteric regulation, right? So Greek allos means other, and steric or steros means shape. So basically what this means is that if you have a ligand that binds to one site, right? It's a different site. It's another site, and it will affect the properties of this of a new site on the same protein, right? And that will bring some kind of conformational change, just like we saw here, right? So there's two different sites, allos. They are not the same, right? But binding of one ligand changes the shape of the other ligand's site. And so this can be positive, can be negative, right? So in this figure, we saw it was positive, right? So binding of one ligand actually increases the chances that the other ligand will bind to, but can be negative as well. And we will see an example of a negative a little later in this lecture. When we have a negative regulator, when it binds, it prevents the binding of another ligand. Can be homotropic, right? So here we'll talk about in the context of hemoglobin. Obviously, oxygen is going to be a normal ligand. Oxygen can be a normal ligand, or it can be an allosteric regulator. So it's both. So these kind of allosteric proteins are known as homotropic. But there's also heterotropic. Also, we'll see an example of that, where here we have a normal ligand, but our regulator is going to be a different ligand. It's going to be heterotropic. So cooperativity is a special case of allosteric regulation, specifically positive homotropic regulation. Okay, so that's the definition of cooperativity. So how is the problem with hemoglobin is solved, right? So remember, our main task here is, let me go back to this figure here. And so we talked about this figure. Our main problem is that, so if we just had a protein that bound one molecule of oxygen with no cooperativity, then the curve will look just like this, right? Basically, you can see at extremely low partial pressure of oxygen, we're going to saturate all the binding sites on the oxygen binding protein, right? Myoglobin. And then you can see the difference in the saturation is the same in the lungs versus the tissues, which means it's going to bind all these molecules of oxygen in the lungs, travel to the tissues, and how much of it will be released? None. None will be released because it's fully saturated in both, right? Now with the low affinity state, you can see there's a problem here as well. All right, so we have obviously it's going to be much more released in the tissue, but not so much more is going to be saturated in the lungs either, right? So the low affinity state is not ideal as well. High affinity and low affinity is not going to accomplish what we want. And it's this sigmoidal curve which will accomplish what we want. It's a transition from low to high affinity state, right? So this must be as high as possible, and this must be as low as possible. And the sigmoidal shape like this, right, will allow us this kind of drop. Sigmoidal. Sigmoidal shape. Sorry, sigmoidal curve. Okay, does it make sense? All right, how is that accomplished? All right, let's go back. So that's accomplished by having not just one protein with a single subunit that will bind oxygen and try to carry it from the lungs to the tissues. Instead, it's going to be a protein, which we'll call hemoglobin, which will bind oxygen cooperatively. It's going to be a tetramer of 2 subunits. So 2 alpha and 2 beta subunits. And each subunit is actually quite similar to myoglobin. So myoglobin, beta subunit, so similar to beta hemoglobin, and this is similar to alpha. And the amazing thing is that I think it was let me double check, 27% or 27 amino acids. And these 3 polypeptides, identical at 27 positions only.
Is that for both the myoglobin and the hemoglobin? They're
All
both
three. They are identical. All they have in common is 27 positions. But they also obviously differ, if you compare them in pairs, right? So, but the 3-dimensional structure, but each subunit is similar to myoglobin. So even though the amino acids are not the same, but you can see that the shape is very similar. So we have alpha helix here, alpha helix here, alpha helix there, and similar alpha helices everywhere, right? So subunits are similar in shape because they need to be similar in shape so that they can carry oxygen, but they need to have different amino acids because they have different function, right? So the function of myoglobin will be primarily to store oxygen in the tissue, in the muscles. The function of hemoglobin will be transport oxygen. And so two different functions require two totally different sets of amino acids. But because they carry the same stuff, the same oxygen, the overall shapes have to be similar. So it's very interesting finding, right? And so the hemoglobin, this is just a general picture. There's not much here to say, but you can see here in dark gray, we have a beta 1, beta 2 subunits. In light gray, we have alpha 1, alpha 2 subunits. And each of these will have a heme. And the sphere here is the iron, right? So there are two heme prosthetic groups. And there's a lot of what we'll see, even though it's not shown here, obviously there's a lot of ionic type of interactions between the subunits. And the ionic interactions will change the shape and will be responsible for cooperativity. All right, so what gives this sigmoidal shape? Basically, there must be a conformational change, right? To get the sigmoidal shape, we must have a conformational change of the protein hemoglobin as it comes from the lungs into the tissues and the other way around. So there are two states. One is what we call tense state, right? T state. Now there's more interactions, it's more stable, and it has lower affinity for oxygen. So this will, which site in the body will this favor? The tissue or the lungs?
The lungs. Is it the tissue? Is that the sigmoidal part?
Well, sigmoidal, you mean like the vertical part of the sigmoidal shape, yes. So lower affinity for oxygen. So where do you want the lower affinity to be? In the lungs or in the tissue?
Okay, in the tissue.
Tissue. Because we want the oxygen to be released in the tissue. Now, R is the relaxed state. Few interactions, more flexible, high affinity for oxygen. And so here we have the lungs.
So this would be the storage part then?
No, this is transport. Transport, so hemoglobin goes into the lungs, adopts the R state, binds as many molecules as possible of the oxygen, goes into the tissue, adopts the T state, adopts the lower affinity for oxygen, and releases the oxygen, again, as much as possible. And so O2 binding triggers this T to R conformational change. So think about it. So now you will have in the hemoglobin, we have 4 sites for oxygen, right? So one way to understand this is, let's say you have a T state, right? It goes into the lungs. Goes into the lungs. Binds to the T state. Right? And then this binding will move it from the T state into the R state. And then obviously we will, once it's in the R state, it gains very high affinity for oxygen. Then we'll have R times 4 oxygens. This makes sense? Once you bind, this is positive cooperativity. T state goes into the lungs, binds 1 molecule of oxygen, changes the shape, and 3 additional oxygens will bind more easily. Now, let's just write fast.
What makes it go from the R state back to the T state?
So that will happen in the tissue, right? So once the oxygens are starting to coming off in the tissue, we go to the tissue where the partial pressure of oxygen is lower, right? As the oxygens are starting to come off, now I don't know if 1 is enough, maybe 2, maybe there are other the problem is it's not a problem, but this is a very simplified explanation because cooperativity is just one of the factors. We think it's the most important one, but as we will see, there are other factors affecting the oxygen binding as well, right? So we'll see the acidity plays a role. We'll see the carbon dioxide plays a role. Does it make sense?
I don't know why I thought higher affinity meant storage in the relaxed, well, for the relaxed state. I guess that's where I'm confused. But I guess it makes
Well,
sense.
the hemoglobin evolved not, it's a very complex structure. You don't need such a complex protein to store oxygen. You store oxygen with myoglobin, which has 1 subunit only. Here, remember, this is myoglobin. This curve, when it gets saturated at very low pressures of oxygen and stays fully saturated, it doesn't matter whether it's in the lungs or the tissues, it's going to hold on to the oxygen. Look, in the tissues, almost 100% of all the oxygen binding sites are occupied. So myoglobin does not need any transfers. All its function is to store oxygen in the tissue.
Okay, that's the difference. Okay, thank you.
Yeah, don't confuse myoglobin with hemoglobin. Myoglobin is a storage protein with a single subunit. Hemoglobin is a transport protein with 4 subunits, 2 alpha and 2 beta. And T2R transformation is for hemoglobin.
Okay, that makes sense. Thank you.
Yeah. And so, so this conformational change from T to R state, we will also see that basically some of the key interactions which are involved is these ion pairs or salt bridges, right? So keep that in mind. Now in the text, they actually have a number of figures which kind of attempt to show you some of these salt bridges which are breaking upon the transition from the T to R state. I mean, I didn't find them very convincing. I think they only get things more confusing as far as what's breaking, where is breaking. So I took them out of my lecture. But if you're interested, you can look at those figures and see for yourself. If you can understand them well, that's great. So, so those are the figures. I'll tell you. Five 9 and 510. Five 9 and 510, which attempt to kind of show you what kind of salt bridges are breaking or forming upon these transitions. All right. So another way to understand. So, but then the question is, all right, what does oxygen binding? How is this accomplished? Right. So so oxygen binds. So what? All right. So here you can see T state, right? 10th state of hemoglobin. So there is this proximal histidine. Remember, on one side it's histidine on the of the heme. On the other side it's oxygen, right? It's going to be over there. So without oxygen, there's no oxygen here. Without oxygen, this histidine pulls iron out of the porphyrin aromatic system, out of this heme, right? And towards the protein. And so that causes some conformational shift. So in other words, this histidine kind of is pulled inside into the protein. So if you draw an arrow, it's like this, goes that way, right? And that will result in a T state. Now, as the oxygen binds, it will bind obviously on the other side of the heme, binds to the iron. Remember, iron has 6 coordination sites, right? 4 within the porphyrin from the nitrogens of the pyrroles, 1 from the proximal histidine, and 1 from the oxygen molecule. So oxygen will pull the iron back, as opposed to being on this side fully. It's now in the middle, even closer towards the oxygen. And that will make this histidine on the helix F, position 8. Right, remember what this means? This means that, remember there are, each subunit has 8 helices, A, B, C, D, E, F, G. A, B, C, D, E, F, G, H. And so this is amino acid 8. So histidine F8, and it will be pulled back out from the protein, and that will result in the R state. And again, the difference between the R state and the T state is the formation of various salt bridges. And you can try to look in those figures if you can understand them a little better. Salt bridges. All right, any questions? So this is the positive cooperativity. This is what oxygen does. Now, it turns out that acidity plays a key role as well. So let me ask you this question, which is written here at the bottom. Does acidity increase or decrease Kd? Just based on this figure, you can see the different pHs, right? So the acidity changes. And as the acidity changes, the curves, the shapes of the curves change as well. What's Kd? Does the affinity, high affinity means low Kd or high Kd?
Low Kd.
Low Kd, okay. And so based on these curves, which curve, using this curve, we can calculate Kd. You should have been able to do this with sapling, and you may have to do this on the test. So keep that in mind. So using these curves, which of these curves will indicate, let's say, higher or lower Kd?
The higher pH, right?
The higher pH will give you lower Kd or the higher Kd?
A higher Kd.
Are you sure?
Well, it would be a higher infinity for the higher pH. Or actually, that's lower pH, right? 7.6.
All right, so remember, Kd is the concentration of the substrate, right? Or the partial pressure of the substrate, oxygen, at which half of the binding sites are occupied on the receptor. And so it will be this. So this will be at high pH, lower acidity, right? Now at high acidity, KD will be this. So the acidity increase or decrease KD then?
Increase.
Increased, correct. So so this will be KD with low acidity. This will be KD with high acidity, right? 7.2 high acidity, 7.6 low acidity. So which means is that if the high acidity increases the KD, then is more oxygen released or less oxygen is released from the hemoglobin?
You say that again?
So if the KD is high, right, then will this result in more oxygen being released or less oxygen being released?
More is released.
More is released. So higher the KD, the lower the affinity, and more oxygen will come off the hemoglobin. And so this situation should be happening in a tissue. And that's exactly what it is. So the tissue has higher acidity than the lungs, and this will contribute to the oxygen release from hemoglobin. So the question is, why do the tissues have high acidity? And that primarily comes from the abundance of this enzyme. There's this enzyme known as carbonic anhydrase. Now, the tissues are actively metabolizing, right? They need food, they use nutrients, they use oxygen that was brought by erythrocytes, they burn the food, and they generate carbon dioxide. So what happens to carbon dioxide? Somehow carbon dioxide, we exhale it, right? We exhale it through the lungs. But the question is, how is it delivered into the lungs from the tissue? Because if it's produced in the tissue, it will just simply turn into gas. Carbon dioxide is not water soluble. Remember, carbon dioxide is a linear molecule. Non-polar. Non-polar. Not water soluble. And that's why there are a number of ways to get rid of it. And one of them is through this enzyme carbonic anhydrase. It will catalyze the reaction between carbon dioxide and water, produced by carbonate ion, and produce H+, which will increase the tissue acidity. It will increase the tissue acidity. And it turns out that hemoglobin affinity for oxygen depends on the pH. We just saw it right here, right? We just saw it right here. And it turns out that when the concentration of protons is high acidity, proton will bind to hemoglobin and stabilize the T state. Stabilize the T state, which will result in oxygen release. Specifically, how will it do it? It will protonate a particular histidine on a beta subunit of hemoglobin, histidine 146, which will form a salt bridge with aspartate 94, and that will release oxygen. And the pH difference, so the pH difference between lungs and metabolic tissues increases efficiency of the oxygen transport. This is known as Bohr's effect, right? Bohr's effect. So which also contributes to the release of oxygen in the lungs. So let's try to understand this using our organic chemistry language. So here's our histidine 146. 146. So beta subunit. Beta subunit. Histidine 146. So remember, histidines are based on imidazole rings. Like so, right? And in the tissue, we're going to protonate this. In tissue. Now, which nitrogen? I think we already talked about this before, but it's a good review. Which nitrogen is going to be protonated? They both have lone pairs.
The top one?
The top one. And what made you decide that it's going to be the top one?
Is it because it's in the p orbital? Or would that be the lone pairs on the bottom? I don't remember.
Well, you don't need to remember. You need to think about it.
Yeah, I think it's the top one, right? Because it's in the, well, yeah, and the bottom has already that double bond. So the top one would be in that p orbital.
So think of aromatic systems. Think of Huckel's rule. Now, is imidazole aromatic? Imidazole is this ring.
No, it's not aromatic.
What does this mean? Huckel's rule. So if you have this number of electrons, when the n is a whole number, then any system with this number of electrons will be aromatic. Remember that from organic chemistry?
Okay, okay.
So, come on guys, participate. So based on this, is imidazole aromatic?
Yes.
So n equals what?
2.
If n equals 2, then you would have 10 electrons.
Oh, sorry. 1.
1. 6 electrons, right? So look at this. The 2 electrons from the double bond, another 2 electrons from this double bond, and 2 electrons from the lone pair. So that makes this ring aromatic. And so this lone pair is busy. If you use this lone pair to protonate it, you will take out 2 electrons from the aromatic system, and you lose a lot of you lose energy, right? The aromatic system stabilize structures. So then I just answered my own question, right? So it's going to be this lone pair which will undergo protonation, because this lone pair is not part of the aromatic system. All right. So let's keep going. Now let's protonate our histidine. plus. All right, and which amino acid is it going to be forming a salt bridge with? Aspartate 94. No, what happened? This new computer did not keep my structures. All right, so if you have that drawn somewhere, I'm just gonna redraw the bottom. Oh, I'm so disappointed. It didn't ask me to if I wanted to keep the my markings. So histidine 146. I'm just gonna redraw this. Imidazole. Plus is going to form a salt bridge, right? With guys, you should draw this because obviously this will not appear in a PowerPoint. So this will be aspartate 94. So positive charge, negative charge. So in tissues, H plus is high. And so this will cause T to R transition. So this will now be known as Bohr's effect.
I have a question about that. I thought it was, I thought the H pluses were stabilizing the T conformation. Do I have those flipped?
Excuse me, I have it flipped.
Okay, thanks.
Thank you for noticing. R to T transition. Got so upset that I stopped thinking. R to T transition. Does this make sense?
Yes.
All right. So there is, that's the acidity. What about carbon dioxide? It turns out that carbon dioxide also interacts with hemoglobin. And remember, so hemoglobin will have a, now, did you record this? Did you write this down? Because when I go to the next slide, this will disappear.
Yes.
Okay. And the next slide is important. So it turns out that hemoglobin and also provides as a means to export carbon dioxide into the lungs. So it turns out 15 to 20% of all carbon dioxide is exported in the form of a carbamate on the amino terminal residues of each of the polypeptide subunits. So if there are 4 polypeptide subunits, right, in hemoglobin, so there's a primary amino group, right, which will be interacting with carbon dioxide. And remember, the way this is going to work is from organic chemistry. Lone pair in the nitrogen will attack the carbon of carbon dioxide. Carbon dioxide. We have O minus and double bond toward the oxygen, and this will be N plus, right? And then water will pick up the proton. Water will pick up the proton, and this will take us to this structure. Okay, and so this proton will be released, right? And this proton actually will contribute to the Bohr effect, right? So more acidity, stronger Bohr effect. And now think about it. So so you form the carbamate linkage, right? Again, what are we having? Stabilization of the T state, R to T. R to T. And and presumably, again, we're initiating some kind of Sol bridge. So think about it. Let's say you have some kind of lysine. Lysine will have the protonated side chain. Remember, lysine has an amino group in its side chain, right? Like so. Plus. And this will form a Sol bridge with the carbamate. Like so. So forms additional Sol bridges. All right, you see, so far we've seen lots of factors. We've seen lots of factors which facilitate oxygen absorption in the lungs and its release in the tissues, right? Do you guys have any questions so far? Okay, so and the final piece, which plays a huge role in the oxygen transport, is this molecule known as 2,3-bisphosphoglycerate. It's shown here. So this portion is glycerate, right? So this amino acid with, sorry, not amino acid, the carboxylic acid with the 2 hydroxy groups, and it has 2 phosphate esters, this phosphate ester and this phosphate ester. So it's 2,3-bisphosphoglycerate. So it's a so it's a charged molecule. In fact, it has what? 5 charges, right? Small negatively charged molecule, and it binds to the positively charged central cavity of Hb, and specifically by doing so, it stabilizes T states. Again, R to T. If it binds, if you use the bis phosphoglycerate. So it's actually present in millimolar concentrations, right, in erythrocytes, and it's a negative. So now, so we talked about, I told you about the allosteric regulators, and I told you that we will see an example of a heterotropic regulator. So this will be a heterotropic regulator because now it's not an oxygen, it's BPG, right? Not homotropic, it's heterotropic. It's a different molecule. And it's also negative as opposed to positive, because if this binds, right, it puts the protein into the T state in which it does not want to bind oxygen. So it's a negative regulator. As it binds, it prevents the actual ligand from binding. And so it turns out that in hemoglobin, BPG binds in the central cavity. Remember, each hemoglobin will actually bind to 4 molecules of oxygen, but it will only bind to 1 molecule of BPG. And you can see here, so if this is T state, right, there's a nice cavity open up in the hemoglobin to accept BPG and to stabilize the T state. Now the question for you, why do you think BPG will go inside? So obviously there's maybe some kind of steric factor, right? Maybe it fits in there sterically. But also keep in mind, what if I tell you that these blue residues, amino acid residues, are usually positively charged amino acids, such as lysine, arginine, maybe histidine. So why do you think BPG will go inside?
Because it's negatively charged?
Yes, it's a small negatively charged molecule, and it loves positively charged amino acid pocket, right? So now in the R state, right? So when BPG is absent and there's lots of oxygen bound to the hemes, then this pocket closes. This pocket closes and BPG cannot get inside. So what we need to understand is this graph here. It takes a while to fully appreciate what's on it, but let's try to get through this together first. And then you may need to actually come back to it a couple of times to go over it by yourself. All right, so this graph shows how BPG helps us acclimatize to living at high altitudes. Or for example, what it does, for example, those of us who are climbers, I don't know if anybody has tried to climb any tall mountains, but when people who climb Everest, right, there's a base camp there. So people spend, I don't know, a week, 10 days. Those of you who have climbed Everest probably know. Spend at the base camp specifically to do what? What do you think happens as you are living on a base camp in preparation to climb Everest?
You're acclimating to the less oxygen that's in the atmosphere.
Perfect. And how do you acclimate to that?
Upregulating your BPG expression.
That's right. You're upregulating the BPG expression. Now, I've always thought, before I actually learned biochemistry by myself, I always thought it's just because these people have more erythrocytes in the body. But that again doesn't explain things very well, right? So again, more erythrocytes, perhaps somewhat, but again, will they release oxygen, right? Will they release oxygen in the tissues? So it's again a question of for them to get oxygen in the lungs and release it into the tissues. So it is mostly due to upregulation of BPG. So if there is no BPG, BPG is 0. The curve is black. It's going to look like this. At very low oxygen pressures, you can see there is no difference between the fact that the hemoglobin is fully saturated. Theta or Y is 1.0 in the tissues. It's 1.0 at the base camp, and it's 1.0 in the lungs on the sea level. Not base camp, but in the lungs of a person living on the base camp. And so there's no difference. Now, at normal, for a person who lives on sea level, this person's BPG is about 5 millimolar in erythrocytes, okay? And that's the blue curve. And so what this person is experiencing is, so the partial pressure in the lungs at the sea level is about what, 13, 12 point something, 13, right? And the partial pressure in the tissue is about 4. It's about 4. And if you subtract the, let's say 0.6 from 1.0, you get about 38% oxygen release, right? Between the lungs and the tissue. So 38% of your oxygen will be released, which is a great accomplishment, right? So now, as you go up and start living at high altitudes, look what happens. Your oxygen pressure in the lungs at high altitudes will be around 7. In the tissue, it's a bit lower. It's 4, right? So the difference is 7 to 4. And hold on a second. 7 to 4. And the difference in oxygen occupancy of the receptor is about 30%. So if you live on a base camp, right? Or I mean, you just climb up there, then only 30% of your oxygen will be delivered from the lungs to the tissues. And how do we restore that back to 38%? By increasing BPG in erythrocytes. So now it's the red curve here, right? It's the red curve here. Again, so we start with the partial pressure of the oxygen on the base camp at around 7, going to 4 here. But now you can see there is a significant difference in receptor occupancy. So which allows us to drop this all the way down there. So basically, oops. Basically, it's going to be that. And that, compared with the partial pressure of oxygen in the lungs at this high altitude, takes us back to oxygen release of 37%. So 38% is close to 37%, right? So again, the idea is increase the BPG to 8 millimolar, you change the sigmoidal curve, and you restore the oxygen release close to 40%. Do you have any questions about this graph? Well, look at it a couple more times. Read the text. If you have any questions, obviously throw them in the discussion on Canvas. Okay.
Dr. Konieko.
Yeah.
So why is BPG not expressed just normally at sea level? Because it seems like it would be a more efficient way to transport oxygen in the blood.
Okay, I've never thought of this. So let's see. So you're saying that we would have then, if let's say here we have this much saturation of the hemoglobin, right? Here we have this much saturation of hemoglobin. Then we will probably have close to maybe 50% oxygen transport. So anybody, anybody has any ideas?
I was thinking about atmospheric pressure.
What about it?
Well, maybe here, isn't it denser higher up there? Or it's I think it's denser down here, and up there it's
Well,
lighter.
the question is, why isn't our BPG 8 millimolar even if we live at sea level, if I understand correctly, right? Why is it at 5? Why not at 8? So what makes 5 better than 8 if we live at sea level? Obviously something does, right? So obviously
I was just thinking like homeostasis, you know, also with atmospheric pressure, it's different here than it is, you know, in high altitudes. So I don't know. It was just
well,
a thought.
atmospheric pressure, you can see the differences here is not much, right? The difference is right there. So whether you have 5 or 8 millimolar BPG, the saturation of hemoglobin by oxygen at the sea level will be almost identical. But in the tissue, it will be significantly different, which will allow you to a larger, better transport of oxygen into the tissue.
Would it be because like even though like you can only use 38%, like it's not worth making more BPG if you can't use all 50% of the oxygen?
You know, there is even with COVID patients, right? So they're on the artificial, they have oxygenation apparatus, right, which will improve their well, lung function. Well, in fact, I should say that this works for people who BPG increases in people who are acclimatizing to high altitudes, but only people who suffer from hypoxia, for example, right? So, for example, people with deficient lungs. So COVID patients would be one example. I never heard about anybody discussing BPG in people with COVID, but in general, you think about it, that should be the case, right? If the lungs are not functioning normally, the BPG should go up because of hypoxia. Yeah, so something keeps BPG at 5 millimolar, and we don't know that.
Would it have something to do with pH?
Say it again?
Did it have something to do with pH?
With pH?
Maintaining the pH?
Yeah, I don't know. Well, let me speak. I'll put something in discussion today. I'll check. I'll look into it. Okay. Good. Very good question. Thank you. So the last thing I wanted to mention is there is a disease. Now there are many mutations in hemoglobin. Now you know there is heterozygous mutations, there are homozygous individuals, right? So let's say, hey, if you have a mutation in both copies of your gene, you are homozygous for that. If you have mutation in just one copy, you're heterozygous for that mutation. So it turns out that if you have a mutation that you inherit from both of your parents, specifically on the beta chain of hemoglobin from glutamate 6, glutamate at position 6 mutated to valine, okay? And so it turns out that that has a deleterious effect. And specifically, what it does, now think about it. So here are your 4 subunits of hemoglobin. This is normal hemoglobin. And this is hemoglobin in sickle cell anemia. And you can see here the beta subunit will change its shape slightly. And specifically, it will make it more sticky. I think the sticky is the key word here, because valine is now capable of all sorts of hydrophobic contact, right? And you can see here, so the beta subunits start to stick to each other to form these long interactions between molecules, forming strands and eventually forming fibers, which will crystallize, will no longer be soluble, will crystallize and change the shape of erythrocytes. So if the normal erythrocytes are discs such as this, right? So these ones will look totally different. They, because of these fibers that crystallize, the erythrocytes will adopt different shapes. And obviously now some of them look like sickles, right? And so that will change the, that will make them less functional. And so these erythrocytes will not be as efficient in transporting oxygen as normal erythrocytes. And usually people with this disease die in their early childhood if they are homozygous for this mutation. In other words, if they inherit this from both of their parents. But the interesting thing, if the person is heterozygous, so if the person inherits this from one parent only, then only 1% of all the people will experience these symptoms of sickle cell anemia. And 99% of those will live normal lives. And the advantage of having, I guess there's evolutionary advantage, especially for people who in Africa who live in areas exposed to malaria, where the malarial mosquitoes present a danger. So that protects them from the parasite. So for whatever reason, this mutation makes these individuals more resistant to malaria than people who have glutamate in position 6 of both alleles. So it just tells you how evolution basically chooses the best possible balanced situation, right? Even though it may endanger certain segments of population to sickle cell anemia, but more people will survive because they will not die from malaria, right? So a very famous mutation. And the final thing I wanted to say is, well, I think we're kind of out of time, but I will say something. There is in the fetus, бабушка, дверь открыта. In fetus, the hemoglobin is actually alpha 2, and instead of the gamma chains, there are, instead of beta chains, there are 2 gamma chains versus alpha 2, beta 2. And it turns out that the gamma chains have high affinity for oxygen than beta chains. So why do you think that is? What advantage? What advantage does it produce for those who are still in the womb?
It ensures that oxygen is picked up like from the mother to provide to the fetus.
Yeah, so the more oxygen will bind to this hemoglobin, and it will be, as you said, picked up from the mother's, more oxygen will cross the placenta and will be available to the fetus for the development. All right, any questions? And I will look up the question about the BPG. And again, I'll be available, start your exam at 11, and I'll be available by email. And that's it. I'll talk to you soon, guys.
Thank you.
Bye.